please have questions. I don't want to just spend another hour lecturing. So, any questions about the exam? Actually, you know what? Let me just give you some general disclaimers based on the emails I've been getting. This exam is 50 questions, multiple choice, based on what was covered in lecture. You should study the PowerPoint slides and not your textbook. Your textbook is a supplement for the exam and has about triple the amount of information in it compared to what we covered in lecture. Like, there's a ton of stuff in your textbook that we did not cover in lecture, even though we technically reviewed the topic. Your textbook has breadth and depth to it. Let's see here. There are 3 versions of the test, and the 3 versions are all the same. It's all the same questions, just different orders or arrangements of the question. Question number 1, everybody should get correct. It's, which version of the test are you taking? If you get that question wrong, I can't help you. That was my gift to you. That's 2% of the test grade right there, knowing what version it is. Yeah, right? I'll take what I can get too. It makes it really easy to keep track of which test was which if they all fill in the question on the Scantron. Version A for A, so on and so forth, or answer A for version A. So those are just some general disclaimers about the test. If you want to show up at 7:30 on Monday to take the test, you have the option of showing up early to take the test if you want more time. We have 50 questions, 50 minutes. Most students take about 20 to 25 seconds of multiple choice question, I've found over the years. We've budgeted 60 seconds of multiple choice question, but you have the option of about 90 seconds of multiple choice question if you decide to show up a half an hour early. So with that kind of bookkeeping out of the way, any questions, comments?
At one point you said that lysosomes and peroxisomes are made in the rough ER. Aren't lysosomes made in the Golgi apparatus?
They are made from the Golgi apparatus, yes. Both lysosomes and peroxisomes emanate from the Golgi apparatus. The proteins that go into those organelles start at the rough ER, are sent to the Golgi apparatus, and then are finished at the Golgi apparatus. The physical organelle directly comes from the Golgi apparatus. Good question. Based on what I was getting asked in my office hours, there's a lot of confusion about introns and exons and pre-mRNA processing. The names are very counterintuitive. Exons are the segments of pre-RNA that stay in. Introns are removed. And if that sounds completely backwards to you, it did to me as well when I was a student. The introns are removed, the exons remain in the pre-mRNA molecule. All of that messenger RNA processing occurs inside of the nucleus of the cell. I had 3 conversations about that in my office hours with 3 separate people, which means some of you are probably wondering about that too. Any other? Oh, right there. Similar, yes. Actually, hold the thought. I should repeat this out loud for the people watching the recording. The questions on Top Hat are similar to the questions that are on the test. Low-density lipoproteins are a solid compound. They are not a liquid. So because if we were to intake a solid, that's categorized as phagocytosis. Uptaking liquids is pinocytosis. Good question. Any other thoughts, comments, concerns? Ooh, I like that. You should have been my study buddy when I was an undergrad. Introns stay in the nucleus. Exons exit the nucleus after the mRNA molecule is processed. That's a good word analogy. I really like that one. Maybe that's why they're called what they're called.
Did you say exons stay in? Yeah. Right, you just said that somewhere. In the RNA. Wait, so they say exons stay in the RNA, but the introns stay in the nucleus.
Yeah, so by introns staying in the nucleus, they're cut out of the messenger RNA molecule and remain in the nucleus. And then the exons remain in the messenger RNA and exit the nucleus. Yeah, it's really easy to get those terms mixed up, isn't it? All right, well, if we don't have any other additional specific questions, let's go through our learning objectives.
Here's some questions.
Oh, in the back. Osmolarity is a way to measure how a solution will influence osmotic pressure. So there are different ways to measure concentration. We could have percent concentration, we could have molarity, we could have molality, but I really dislike that unit of concentration, and osmolarity. When we look at osmolarity, molecules that dissolve into multiple particles are going to have a higher impact on osmosis and osmotic pressure, and will give you a higher osmolarity, or will influence osmosis more. Molecules that don't dissociate or break apart into multiple ions when you dissolve them aren't going to have as much of an impact on osmosis and won't have as high of an osmolarity. So we're not going to have math on the exam. So I'm trying to explain this without math. When we think of osmolarity, it's a unit of concentration that tells you how the chemical is going to influence osmotic pressure. I think the examples I gave in the notes included sodium chloride and glucose and probably calcium chloride. Those are some common solutes. Glucose, when you dissolve it, remains as one particle and doesn't have that much of an influence on osmosis because it remains as one particle when you dissolve it in liquid. Sodium chloride, when you dissolve it in liquid, breaks apart and forms two separate ions and has more of an impact on osmosis because it has the two separate ions when you mix sodium chloride with water. And then there's calcium chloride. When you dissolve calcium chloride in water, it forms three separate ions and has triple the effect on osmosis compared to the same amount of glucose. So when you think of osmolarity, it's a way of taking into account the number of ions or particles something separates into when you dissolve it, and their impact on osmosis. Any other questions, comments, concerns? No. So I'll repeat that out loud for the recording's sake. No, you do not need to look at diagrams that identify things on the exam from diagrams. That we hit up during lab. We really emphasized identify, identify, identify during lab. Here in the lecture portion of the course, we're strongly emphasized the application and the function. So you should be able to read the name of the structure and then know the function of the structure. Good question.
Is the exam going to go in depth as in like with the proteins and stuff that things like myosin, moving, chromosomes and anaphase and stuff, that kind of stuff?
We don't necessarily, it's fair game. If it was covered in the lecture, it's fair game for the exam. With that said, it's only a 50-question exam. And if I spent a lot of time asking very nitpicky questions about cell cycle, that's using up a lot of my real estate in terms of my exam. Then that means I can't ask questions about other stuff that I covered. So when I was doling out questions for the exam, I did not succeed perfectly. This is, an imperfect process. I tried to have the same number of questions on the exam per lecture period. So we spent a day on introductory stuff. We spent, officially in the course calendar, there was a day on chemistry. We had a day on cell cycle. We had a day on protein synthesis. So there should be about the same number of questions on each of those big topics on the exam, based on how much lecture time was scheduled for each of those topics.
Is there a big difference between codon and base triplet?
Columnar and base triplet?
No,
Codon. Oh, good question. So when we think of a codon versus a base triplet, let's start from the top. So I'll back up a little bit, then I'll get to your question. That was a protein synthesis question. And for protein synthesis, the big picture, the kind of the umbrella, the framework you need to view everything in is we go from we go from DNA to messenger RNA to the protein. Now, in our DNA, we have our genetic code. And that genetic code, when it's in the DNA format, the deoxyribonucleic acid format, every 3 adjacent nucleotides is called a triplet. But then we take the DNA and we turn it into messenger RNA using base pairing rules. And when you go from DNA to RNA, the triplet in the DNA becomes a codon in the molecule of messenger RNA. So kind of like how I had you do in lecture yesterday. Was that really yesterday? It seems like a really long time ago. Anyways, during lecture yesterday when I gave you the sequence of DNA, and then I had you transcribe it to messenger RNA, and then use the codon cheat sheet to translate it to amino acids, there's going to be a question just like that on your lecture exam. I'm giving you a segment of DNA, and I'm giving you a codon cheat sheet. No, you do not have to have the codon cheat sheet memorized.
So codon refers to the nucleotides in the mRNA?
Correct. 3 adjacent nucleotides in the messenger RNA that code for a specific amino acid. Or maybe we could also say a stop codon as well. And really, you technically don't have to memorize any of the codons because I'm giving you the codon cheat sheet, but the one that's really worth committing to memory, out of all 64 of them, the one that's worth committing to memory is AUG, is the start codon. So when you're scanning along your segment of RNA, you should not add any amino acids until you find an AUG. And since this is a study session, and hopefully we're sitting a little bit closer, I have an example for you. Let's say we have a segment of letters, and it says the big fat cat. They're all 3-letter words. Each 3-letter word, if I have the correct frame of reference, has a very specific meaning. Where I can say the big fat cat. Kind of like codons in your segment of messenger RNA. And the reason the start codon is so important is it gives you the frame of reference. When do you start reading off every 3 nucleic acids in the messenger RNA? If our start codon was shifted 1 to the left or 1 to the right, the codons downstream would all be changed significantly. And that would significantly change the amino acids in your protein and change the final product. So if I take our frame of reference and shift 1 to the right, I have heb, igif, ek. It means something completely different as opposed to the big fat cat. And the same is true when we're reading the codons in the messenger RNA. That AUG, the start codon, is such a vital codon because it tells you what all the other codons are going to be. Yeah, absolutely. So let's draw a picture. Hopefully I can find a marker that works a little bit better. This one's probably not, but let's see. So if I had A, G, U, A, G, C, A, U, G, A, U, C, C, G. As you're trying to figure out when to start adding amino acids, what you should do as you're scanning this is look at the first three and go, oh, not AUG, not AUG, not AUG. And keep scrolling, looking at 3 nucleic acids at a time. And still not AUG, still not AUG. Whoa, there's an AUG. There's my frame of reference. And now every 3 codon, or every 3 nucleic acids afterwards will be a codon. Bingo, exactly. So when you're trying to find the start codon, you will advance 1 nucleic acid at a time to look for the AUG. Once you find the start codon, the AUG, then it's every 3 nucleic acids at a time.
So since the set like 3 letters, would you stop after the AUG?
Ooh, excellent question. What if you have a remainder, an incomplete codon, so to speak, at the end? If you have a less than 3 nucleic acids at the end of your messenger RNA, there is no amino acid for that. You need to have all 3 nucleic acids to make a codon. An incomplete codon will not code for a segment or will not code for an amino acid. It's all or nothing with codons. Any other questions? Yes. Ignore all nucleotides in the messenger RNA before the start codon.
And then do you know when the codon is done, will end up with one that's stopped from the codon cheat sheet?
Yes, so you will stop translation when one of 2 things happens. First, you can hit a stop codon. And if you get to a stop codon, well, you should probably stop adding amino acids. The other thing that could potentially happen is you run out of nucleic acids. If you run out of messenger RNA, stop adding amino acids.
This might be a dumb question. But so when you're translating this, like with the little cheat sheet,
Yep.
do you like find an A, find B, find a G, or do you do the opposite?
Oh, so when we're actually going through this process, you first find the first nucleotide of your codon on the cheat sheet. The cheat sheet I'm providing for you has first nucleotide on the left, and then second nucleotide is on the top, and third nucleotide is on the right. So let me pull that up on the screen for you. This is the codon cheat sheet I gave you for your exam, the one I included on your test. Maybe I can make that a little bit bigger for you. I like this codon cheat sheet because it's crystal clear. It shows up really well in a black and white photocopy. So if I was working with the AUG start codon, I would have the A, and then I'd go up to the top and find U, and then I'd end up in this quadrant, or this cell. And then I can go, oh, AUG codes for methionine. And that's when I can start adding amino acids. This particular cheat sheet does not have the third column. The one I included in your lecture presentation did have the third column on the left-hand side. So once you find the correct cell to look at, you can just look at the U, C, A, and Gs. You can read through the 4 options within that cell to find the third nucleic acid that you need for your codon.
So if you give us a codon in DNA, that's when you have to transfer or like switch letters, but if you've already given it to us in mRNA, that would just go right.
Yes, yes. So if I give you a segment of DNA, you first need to take the segment of DNA and transcribe it to messenger RNA. And then once you transcribe it to messenger RNA using base pairing rules, use the cheat sheet to figure out the amino acids. If I give you the messenger RNA sequence directly, you can skip straight to the codon cheat sheet. As a rule of thumb, if there is a thymine or a T in the genetic code, is it going to be DNA or messenger RNA? DNA, because DNA will have the thymine. And if it's a sequence that doesn't have any thymines in it but has a bunch of uracils in it, it's probably going to be the messenger RNA sequence. 1 second. I'll get to you in a moment. We had somebody up in front first. Yeah, you raised your hand. All right, never mind.
So if you give everyone your cell, you can skip to that reading that table?
Probably. Read the question. If there's uracil or U present, it is a kind of RNA. And there are 3 kinds of RNAs we talked about. We talked about messenger RNA, ribosomal RNA, and transfer RNAs. So read the question. Make sure that the question is talking about messenger RNA. And if it's talking about messenger RNA and asks for the amino acids, then you can skip straight to the codon cheat sheet. I'm loving all the questions we're getting so far. I've been to review sessions where it's just been nothing. Thank you to those of you asking questions.
Sorry, I have one. So those are just like the letter words. So like, is that the like abbreviation?
The 3-letter symbols for the amino acids?
No, the ones like after it says.
Yeah. So like, for example, if you look in the lower right-hand corner of the screen, if you had a codon that was GGG, that codon would encode for an amino acid called glycine. I don't expect you or want you to memorize the amino acid abbreviations. For the sake of space on your exam, I chose to use 3-letter abbreviations with the codon cheat sheet and the options on your multiple choice question. There's also 1-letter abbreviations as well. We're not using those either. In the back.
Is it Scantron that you're talking
Yes. Yes, we are using Scantron exams. I had a meeting this morning with LTS about how Scantrons are graded in this organization.
So yesterday in lecture we had the protein processing and secretion. Can you just like briefly run through that process?
Absolutely. So when we secrete a protein, it's going to be made in the endomembrane system. So generally speaking, a protein that leaves the cell, that's exocytized or that is secreted. Leaving the cell, exocytized, and secreted all mean the same thing. If a protein does that, it will be synthesized by a ribosome that is attached to the rough endoplasmic reticulum. And then that protein goes from inside of the rough ER to the Golgi apparatus. And it's transported from the rough ER to the Golgi apparatus. At the Golgi apparatus, it's modified a little bit more. And from the Golgi apparatus, it's secreted from the cell. Hold that thought. Would you like some more elaboration on that process? Or was that what you're looking for? OK, because I could talk for probably an hour about proteins modification and secretion.
Say, can you please repeat that?
All right, I'll repeat it again. When a protein is exocytized or secreted or leaves the cell, and that all means the same thing. I'm saying the same thing different ways. When that happens, the ribosome that initially translates the protein will be attached to the rough ER. And that protein, as it's being made, will be pushed into the rough ER by the ribosome as the ribosome is making it. Once the protein is made, it goes from the ribosome it goes from the rough ER to the Golgi apparatus. At the Golgi apparatus, it's modified a little bit more with the different chemical environments inside of the membranous folds, and it will then be secreted from the cell. Lysosomes and peroxisomes come from the Golgi apparatus. So the proteins are going to be made at the rough ER. They'll be sent to the Golgi apparatus. They'll be modified at the Golgi apparatus. And depending on what the proteins are, they might be secreted from the cell Or there might be a segment of the Golgi apparatus that buds off and remains in the cell. If it buds off and remains in the cell, it'll probably be a lysosome or a peroxisome. In the back.
You said that smooth ER and peroxisomes both detoxify alcohol and other drugs. Are those both share a main function?
That is just a function they both happen to share. Difference would be that the smooth ER synthesizes lipids. Peroxisomes do not synthesize lipids. All right, what's the difference between a lysosome and a peroxisome? You could think of the lysosome and the peroxisome as cousins to each other. And these are organelles that have similar functions in many ways, but they go about their similar functions in slightly different ways. They both are going to be focused on breaking stuff apart, but they have a different emphasis. Lysosomes are going to primarily focus on breaking larger things apart. So if we have a bacterium that's been phagocytized, that bacterium, after it's swallowed by the cell or phagocytized by the cell, will have a lysosome fuse with it, and then the lysosome releases digestive enzymes and chops up the bacteria. Peroxisomes are going to focus not so much on bacteria or things that have been brought into the cell. Instead, peroxisomes are going to focus on breaking apart pieces of the cell that need to be removed. So defective organelles, defective proteins. And when we think of, and this is just a little bit of, cell history or molecular biology history for you, peroxisomes and lysosomes, in terms of organelles that were discovered, they're still kind of the new kids on the block. They weren't differentiated from each other until fairly recently in scientific history because they look so similar underneath a microscope, and they have relatively similar functions to each other with slight nuances that separate them. There was a question in the back.
There was a question that said that what organelle would be like an enzyme-filled sac or something, those lysosomes. How would that be different than a peroxisome?
An enzyme-filled sac is a good description of both a peroxisome and a lysosome. So if that question was on the exam and peroxisome and lysosome were options, that'd be a really poorly worded question because it would have two correct answers. Absolutely. A polyribosome is multiple ribosomes translating the same piece of messenger RNA at the same time. I think the example I gave in class yesterday was multiple people reading the same book at the same time, where everybody's at a different point in the story. With a polyribosome, those multiple ribosomes are at physically different spots on the messenger RNA, and they're all scrolling along in the same direction.
I just have a question. It might be going too in-depth that we don't even know about yet. Polyribosome?
Yep.
Does each ribosome start at the start codon and then move?
Yes, they do. Each ribosome starts at the start codon. Another analogy I've used for polyribosomes is a cafeteria buffet, or a buffet, where everybody starts at the beginning, picks up an empty plate, and then they walk the same direction through the buffet line, putting food on their plate. You could have multiple people at the buffet table at the same time, but they're all at a different stage of putting food on their plate.
So do polyribosomes happen after termination occurs?
When we get to termination, the ribosome will release from the messenger RNA. So if we were to think of this front table as a segment of messenger RNA, and the start codon is right here, we could just keep adding ribosome, add another ribosome, add another ribosome, and the ribosomes would scroll along the segment of messenger RNA, and then when the individual ribosomes get to the stop codon at the end, that individual ribosome by itself terminates and separates from the messenger RNA. But the other ribosomes that have not yet gotten to the stop codon will keep translating until they get to the stop codon. Polyribosomes are related to termination in that if the termination sequence is really close to the start codon, you have fewer ribosomes that can attach at the same time. If there's physically a lot of space between them, between the start codon and the stop codon, then there's physically room for more ribosomes to attach to the messenger RNA. It's kind of like a short buffet table versus a long buffet table. Hold that thought.
So do the polyribosomes make the same proteins?
They all make this, every ribosome within a polyribosome complex will make the same protein because they're all reading the same piece of genetic code.
So you said like when the ribosomes get to the termination point, like the stop point or whatever, what happens after that?
The two subunits, the large and small subunits, just separate from each other. And amino acids will no longer be added to the polypeptide, or the primary sequence of the protein, in red.
I just want to double check and make sure this is not on the topic of RNA. When you go over organ systems, you said we don't have to identify anything with other functions, so if I say skeletal system, we have to know that it supports and moving of the body, not that it's both.
That is correct. So with organ systems, for lecture, we're big on function in this class. Know the functions of the organ systems, not the individual organs of the organ system. Yes, we're going to focus on function of organelles, not identification of organelles. Yep.
Can you explain how the tRNA frames the amino acids in your ribosome?
I could try doing it with words again, but I'll admit I'm not that gifted with the English language. I'm going to play a little video segment for you that I think does a really, really good job, better than I can do with just words, because it's words plus pictures, live motion pictures. And of course, the audio's not working. No surprise there. So I'll just scan ahead. So at this point of the video sequence, in blue is a ribosome. In green are transfer RNAs. And the little transfer RNAs are bringing amino acids into the blue ribosome. There are 61 different transfer RNAs in our cell. Each transfer RNA has a different amino acid, in this case shown in red, on it. Each one of our 61 transfer RNAs have different amino acids, within the 20 amino acids to choose from. And as those transfer RNAs are brought into the ribosome, they enter from the right-hand side. The anticodon on the transfer RNA will base pair with the codon on the messenger RNA. And if they match each other, the tRNA molecule moves to the middle of the ribosome, drops off the amino acid, and then the protein moves out of the top of the ribosome. And then the empty tRNA exits from the left. So a tRNA carrying an amino acid enters, base pairs its anticodon to the codon, drops off the amino acid, and then an empty tRNA exits from the ribosome. I want to encourage you to watch this video segment when there is audio, or at least you have working speakers to work with. The narration of this is phenomenal. Does that answer your question about how tRNAs drop off amino acids? Okay. Excellent. Other thoughts, comments, questions?
When does the tRNA use ATP?
The tRNA will use ATP when an empty tRNA has a new amino acid attached to it. Good question. Correct. The anticodon of the tRNA molecule matches up with the anticodon of the tRNA molecule matches up with the codon in the messenger RNA. Both. Because a tRNA molecule is designed to have a specific anticodon in it. And it's also designed to have a very specific amino acid attached to it. So if you change the sequence of DNA, if you change that base triplet, you end up changing what the codon is in the messenger RNA, and then you change which tRNA brings an amino acid, and you ultimately change what the amino acid is in the protein. So to go back to our genetic code, if you take the genetic code and you add one nucleotide or you subtract one nucleotide, that's called a frameshift mutation for those of you who've taken genetics. And those frameshift mutations will change all the downstream codons. And instead of having the big fat cat, you could go from something that has meaning to something that is completely incorrect. For the record, you do not need to know the term frameshift mutation for your exam. It was not covered in lecture. It just kind of slipped out. Sorry about that. Any other questions, comments, concerns?
What's the difference between mRNA and tRNA? The tRNA is attaching to the mRNA?
tRNA does attach to messenger RNA when their codons and anticodons match up with each other. To really summarize it and give you like a statement that really summarizes the functions of the tRNA and messenger RNA, messenger RNA carries the genetic code out of the nucleus. tRNA adds amino acids to the growing protein. Messenger RNA carries genetic code out of the nucleus. tRNA is going to add amino acids to the growing protein.
So the code that the mRNA brought out is nucleus.
Yeah, based on the codons in the messenger RNA. So, any other questions? Let's switch topics. Questions about something other than protein synthesis, because this exam is about more than just protein synthesis.
For the radiology scans, do we just have to be like kind of familiar with them, or do we have to like apply them to like
No application of radiography or radiology scans. This is not, it hasn't been covered in that kind of detail yet. So just at a very general level, you know, X-rays use, well, X-rays. Or a sonogram uses sound waves. A PET scan uses radioactive glucose. In the back.
Are you going to focus a lot on the specific like monosaccharides, disaccharides, polysaccharides?
I would love to ask lots of nitpicky questions about the carbohydrates, but this exam is too short for me to justify asking those kinds of questions. Right there.
For the concentration gradients and the movement of like solids and stuff,
Yep.
the only one that requires a membrane is carrier mediated, or does osmosis also require?
Osmosis also requires a membrane because you need to have a concentration gradient for the water or a membrane for the water to move across, so that there can be some kind of concentration gradient to power osmosis. So yes, osmosis requires a membrane, and facilitated carriers require a membrane.
Your chemistry review from like the very beginning is like super long.
It's all fair game, But that was just an information-dense lecture. Any other questions, comments, concerns? We have about 5 minutes left. In the front. I plan on curving the exams so that the high score becomes the 100% mark. So if the high score is 10 points or 10 questions correct, 10 questions becomes 100%. If the high score is all 50 questions correct, then the high score is 50, or 100% becomes 50 questions.
Should we know the process, like the whole process of the G proteins, or do we just need to know?
No, do not focus on the nitty-gritty details of G protein coupled cascades. G proteins take a signal from outside of the cell that can't cross the membrane and amplify the signal on the inside. And they use cyclic AMP and calcium as secondary messengers.
Can you just talk about briefly, like kind of a summary of like vesicular transport? Just like how it works?
Sure thing. So with vesicular transport, there are going to be substances in the cell that we want to have isolated, chemically isolated from the cytosol of the cytoplasm. And those substances will wrap up in a bag of phospholipids called a vesicle. And if we take that vesicle and move it around in the cell, we can send it out of the cell. That's called exocytosis. If we're bringing something into the cell with a vesicle, we call that endocytosis. And when we have exocytosis, there's really only one flavor of exocytosis, something leaving the cell. For endocytosis, we have 3 variations of endocytosis we covered in lecture. There was phagocytosis, bringing a solid substance into the cell, pinocytosis, bringing a liquid substance into the cell, and then receptor-mediated endocytosis, which is bringing very, very specific molecules into the cell after they attach to the outside. Any other thoughts, questions, comments? A question I was asked probably 4 or 5 times this morning during my extended office hours was, how long should I study for the test? I don't have a good answer for you on that one, because you, as an individual student, are different from your neighbor, are different from me. You know, we all are unique individuals that have unique abilities to learn, retain, and process information. I can give you some general answers, though, to that question I was asked a lot. In general, students that get A's in anatomy and physiology, the average A student studies between 12 and 16 hours a week for anatomy and physiology. For me specifically as a student, I was never done studying for my exams when I was an undergrad. I never felt like I arrived at the point where I know the material so well I can go do something else. Usually what ended up happening for me personally was I ran out of time to study anymore. And I just kept studying and studying, took the test, and then went, oh, okay, it's done. Time to start immediately studying for the next test. Based on our discussion today, most of what's going to be on the exam, because we spent about a half an hour talking about just protein synthesis. And there's a lot more than just protein synthesis on the exam. But that's what most of the questions were about, and that's okay. That we're asking a lot of questions. I wanted to answer your questions as the students. I can give you a list of topics students struggle with traditionally. With protein synthesis, students struggle with taking a segment of DNA and using it to figure out what the amino acids are. That's one of those infamous topics in biology. You know, it's one of the hurdles. Another topic that's traditionally a point of struggle is how we take sugar, glucose and use that sugar to make ATP, or cellular respiration. And that's what we're covering in class tomorrow. That's one of those tricky topics that there's a lot of memes complaining about.
I covered cellular respiration before, and the questions usually revolve around like net gain and stuff, and like what was used and what was produced. Is that what the questions are?
Oh yeah, I am not reinventing the wheel with cellular respiration. Although I will say one of my differences compared to how it's traditionally been taught is I call it the citric acid cycle as opposed to the Krebs cycle, because that citric acid cycle is a more descriptive term. Let's see, other topics students traditionally struggle with. Protein synthesis is a tricky one. Cellular respiration is a tricky one. Organelles students traditionally haven't struggled with at this level. I would say when I was a middle school science teacher, organelles were like the hard topic I taught my students. Knowing body systems traditionally hasn't been very hard for my college students. You know, the urinary system helps you make urine. It filters the blood. It's not something that my students struggle with at the college level. I would say that the two big tricky topics are protein synthesis and cellular respiration. Depending on how much you remember from the chemistry class you took as a prerequisite, the chemistry review portion might be the easiest thing ever, or it might be causing you a lot of anxiety. So we have time for one last question. One last question. All right. Well, I prepared a presentation for you for this review session. I am glad I did not have to pull it out, but I'll show it to you, and I will post it online as well for you. Let me get that out for you. If we pull up Top Hat, just to guide the discussion, I had prepared this presentation on just what my specific goals were for you, what I want you to specifically know about each specific thing we covered in class. The 3.1 or the 2.4 or the 3.2, that tells you the segment of the textbook I pulled the information from. And then I have the specific learning goals I had for you. So I'll post this on Canvas. If you're looking for maybe some other way to guide your studying over the weekend, this is just another tool in your toolbox that you have. Good luck on your exam. Good luck studying, and I will see you tomorrow. Well, we talk about cellular respiration.